Okay, so almost, I guess. Good afternoon. How are you? Good? All right. So anyway, last week was kind of chaos. I don't know if you read my email. So in the middle of lecture, just sound basically stopped recording. Good thing I was prepared with my second recording, so I had to extract my sound from my iPhone video, which was a lot of work because it's 5 gigabytes, and then I can't transfer to my PC, and I had to do everything in a Macintosh, and it's just several hours of work at night. Uploading finally. So if you are relying on the video lecture, the last week one is basically, it's not high quality sound, noisy, but you know, what can I say? That's what I basically, the only one I can. So anyway, hopefully so I'm paying attention to my sound today. So if you notice anything, just let me know, and I'm gonna do something on that. But anyway, so hopefully the IT person came and checked everything, so it shouldn't be too much a problem. Okay. Oh, that's an old one, but I want to show you the. So just want to, okay. So light intensity and light quality lecture number 1. So this is really good lecture. You like it. But a lot of terminology. So one of the goals you achieve in this lecture and next lecture, you get familiar with the terminology. All right. So that's one thing. So light environment, lecture number 1. So today's lecture is more about light intensity, radiance. And then Wednesday lecture is more about light quality, colors of light, and how plants respond to that colors. So that's Wednesday lecture. And then there are multiple reading materials. It's all loaded in your D2L. Some of them are web magazine. Some of them are, library copies for your use. 4 reading materials. That's quite a bit. But the first 2 are for you. And third one and the fourth one are optional. And then the fourth one is something I really encourage graduate students to read if you're a graduate student or taking this course as graduate student level. So that's something I want to recommend. So the first one is Growers Magazine. It's written by a professor from Michigan State University. And this daily light integral concept, we basically understand at the end of this light intensity lecture. What is daily light integral and why it is so important? And then the second one is general reading, why light is so important, why we have to manage light intensity and quality in a greenhouse. So that's the second reading. And that is coming from a little booklet called Lighting Up Profits. Nice title, huh? Lighting Up Profits. So we are actually revising this one and then updating the contents. I will be one of the chapter's author this time. But anyway, so one of the chapter is that general intro to the light environment. So I thought, particularly for undergraduate students, it's good. Oh, one more thing, yeah. So that, right? And yeah, and then the third one is Growth Chamber Handbook. I use this handbook a lot in this course. And then the first chapter is the radiation. That's a light environment. However, there are several typos in that article, even though it's very useful. So I had to correct that. So this is my sort of copy, but everything else is online available. So next time you see chapter cited from this book or booklet, Growth Chamber Handbook. That's going to be online material. And then all the chapters actually online. And if I can show you, yeah, so this is a website, Plant Growth Chamber Handbook, and then all the chapters are downloadable from this website. And I didn't link to this radiation chapter because, again, it has some typos. So I collected and then I uploaded that corrected version in my D2L. Okay? All right. Okay, and then that's the third one. And the fourth one is a graduate student one, so the plants and microclimate. A lot of equations, but this is a good one. Radiation chapter. Many pages, 20-some pages, but it's a good to read. All right, so that's the reading material. And so, oops. Okay, so what is light? Okay, so what is light? So we're going to start with definition. So light is a part of radiation, or a kind of radiation. And then radiation is, what we understand is basically having a wave type of characteristics. So the energy packet, packet containing some level of energy moving in space, or going through or travel through the space, right? And we characterize the radiation using how many number of waves per meter, or wavelengths. And what is wavelength? So if you look at this is a red radiation, red light. So it's having a relatively longer wavelength. So wavelength is a distance between peak and peak. Okay, so the one cycle. So that's the wavelengths. So if you have a short wavelength, that means more number of waves. So it has a much higher energy level. Right? And then the longer wavelengths, it goes much slower, right? So the lower energy level. Again, the radiation has wave characteristics, and then also energy packet, so they're having energy. So when it hits the receptor, and then transfer electrons, and then increase the receptor's energy level up. So that's what it does. Okay, so this equation is a very simple equation showing the relationship between wavelengths, so the one cycle length, and also some important characters like constant. Planck basically defined this constant, and then speed of light because it travels through the space. So basically those two are constant. So the numerator, so this is a value, constant value, right? Two constants divided by wavelengths. So that means this is energy level per photon. So one packet of light or radiation, how much energy it has, and then that can be defined basically using wavelengths. If you know the wavelengths, you know the energy level of the packet. Okay, and then if wavelength is longer, that means energy level is lower because so divided by wavelengths is energy. So if you have greater or longer wavelengths, then you have smaller energy level. Okay, so so that's the equation, and then this particular equation. This particular equation uses meter in the unit. So usually the wavelength is expressed in nanometers. So you need to consider that when you work on this equation. So you insert nanometers as meter, right? So 0.0000 some meter, and then you can compute the energy level, and unit is a joule per photon, one particle energy level. You can find out. All right, so the longer the wavelengths, and then lower the energy level is. And then, so more practical definition for you, maybe if you say light, and that means something you can see, visible light or visible radiation. So that's more, practical definition. And what is visible radiation? What is visible light? And which light you can see and which light you can't see. For example, UV light, you may not see it, right? But red light you can obviously see. Green light you can obviously see. That's why traffic lights work that way, right? So we define radiation using wavelengths. So certain range, wave band, we call this radiation as UV radiation or infrared radiation or visible radiation. So what is a visible radiation's wave band, which is generally defined 330 to 730 nanometers. So nano is, again, if you remember, prefix of SI unit, 10 to the minus 9, yeah. So it's very, 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 very small, right? Okay, so visible light or visible radiation. This is just a presentation so that you can see different colors. Starting from 380 up to that level, you really don't see in your eyes. So that's why it's starting with a very, very dark purple. And then gradually, you start seeing it. So that's the threshold, 380, and then going into blue range, and then going into green range, and yellow-green. And this range, you see really well. You can sense green, yellow-green very well in human eyes. And then yellow and orange, and then red and dark red, and you can't see anymore. So that's about 780 nanometer. Yes?
I'm just wondering, is this like just how our eyes work, and that's why we can see them?
Yeah.
Okay.
Yeah. So the receptors we have sort of get the signal out of this range. So that's, I don't know which one first, but anyway, so that's how it works. So 380 to 780, visible radiation. All right. And then, again, light or visible radiation is part of the much, wider range of radiation. So if you say radiation, there are many more different radiations other than visible radiation. So this presentation is showing you in terms of wavelengths, nanometer, or this one is a meter in a unit, where the visible radiation is. So this is a wavelengths, and 10 to the power of minus 10, minus 9, minus 8. So this is getting longer radiation wavelengths, right? And then visible radiation is sandwiched between UV and infrared, IR, infrared. So longer wavelengths than visible radiation is called infrared. And then beyond that, there is microwave and radio wave. Shorter wavelengths, shorter side from the visible radiation is UV. Okay, again, UV is not visible radiation. And X-ray and gamma ray, and much more powerful, high energy radiation in there because it's very short wavelengths. Okay. So 380 to 780, again, color presentation. And then I also show the energy level right here, corresponding to some of the wavelengths. So you can see the energy level per mole, in this case, not per photon, it's 1 mole, shown in here. So 3.0 times 10 to the power of 5, fifths joule per mole. So how do you find the per mole energy level? It's very simple. So you plug in the wavelengths in this equation, right? Having Planck constant. So this is a constant speed of light divided by wavelengths, and that gives you energy level per photon, and then multiplied by number of photons per mole, which is basically Avogadro's number, 6.02 times 10 to the power of 23rd. And then it gives you number of photons per mole. So by this equation, upper equation, and then this number of photons per mole, then you get energy level per mole. And then if you are converting some of the data, energy-based, expressed, the radiation level into moles per square meter per second, then you need to know this, relationship for every single nanometer. Okay, every single nanometer. You have a question? Still, you have a question?
Under what wavelength do you see the color white? Because you can't make white with those colors, right?
Yeah, so what you call primary colors,
Yes.
3 primary colors, and then if you mix that, blue and green and red, if you mix that, then it looks white. So that's white light. White light is basically broad, spectrum containing 3 primary colors. So there's no white wavelength. There is a red range, so the waveband for red, waveband for green, generally speaking. So for example, if you go back, here, yeah. So here it's about green, right? And then this is like a bluish wavelength. Although it's not monochromatic, it's changing. But because of the convenience, we define, okay, let's call 400 to 500 as blue, 500 to 600 as green-ish, and then 600 to 700 as red. So just a practical definition, but it's actually gradation, right, in that, because it's how we sense the colors in our eyes. All right, so there's no white. But if the lights include those, then you see white light, right? So white light coming from my projector, right? And then that is obviously having 3 components in it somehow. Maybe more, spikes, but range of that prime colors covering some blue, some green, some red, therefore it looks white. Make sense? Okay, good. All right. This doesn't show in my... Okay, I'm going to skip that, but if you look at the PDF version or handout version, it has a chart showing energy level relative to the wavelengths, right? So that's what, and then it's basically it's almost like a you can see the relationship, but basically it shows a inverse relationship between energy level and wavelengths. Okay, I need to remember that it's not really good at showing Excel in here. Okay, so importance of light for plants. This is a summary, what kind of effects and what kind of actions light would cause on plants. And number 1 is a thermal effect. It warms up the body of the plants, increasing temperature, right? So the relationship between plant body temperature and radiation and surrounding air temperature and all that relationship, we're going to learn in different lecture talking about energy balance of the plant. Photosynthesis obviously is very important, right? Plants take light energy and produce carbohydrate using photosynthesis. And also plants use light as a signal to, maybe to stretch or maybe to start flower. So those are the morphogenesis and morphological influence of the light. So plants use light as energy, and then also signal, all right? It's kind of cue to, make action. And some kind of light, if you are talking about light in a much wider definition than gamma radiation, like a shortwave radiation, like that, could induce a mutation, okay? So those are the sort of big summary of light inference. So in my talk, I'm going to focus on photosynthesis because that's what you learn in introduction biology and some of the plant physiology intro course, and then you probably the most familiar, in terms of plant response to the light. So what the general response of photosynthesis regarding light intensity, irradiance. So the light intensity, irradiance, and fluency, those are the synonyms. And then I tend to use, and following horticulture engineers, I tend to use light intensity. It's a simple way to express. But some people means by light intensity as the intensity of the light source. Okay, so there is some confusion there. But in my lecture series, I would stick with light intensity. And then if I say light intensity, that means irradiance. That means the light received per target surface. Okay? All right. So, and then I'm going to talk about how to quantify light intensity. So that's two things you are going to learn in here under photosynthesis. So measurement, right? So that's why we started with unit last week as a first lecture. So measurement of light. So what kind of measurement we use? There are generally 3 measurements used in references you might find in plant sciences, horticulture. However, as you learned last week, the last one, illumination unit, lumens per area, which is based on candela, which is the light flux coming from the light source. So those are the candela-based unit, lux and foot candle. Lux is per square meter base, foot candle is per square foot base. Okay, so those are not appropriate units for using plant science area study. Okay, so you have to use either radiation or photon flux that is expressed using number of quantum or quanta per square meter per second. All right. So radiation is, this is an energy unit. So watt is the derived unit, if you remember, and then it's basically joule per second, energy level per second. So watt per square meter meaning energy going through a square meter of area per second. So the light is continuous, right? So it's not just, flashlight. So you can measure per second rate. And then we usually express per second rate using per second, right? And then energy unit, watt per square meter, okay? Because second is included in the watt. Number of photons, moles or micromoles, often because the unit or quantity is very small, so you need a prefix of micro, so expressing micromole per square meter per second. And the meaning is the same. Number of photons expressed using mole instead of counting all, right? So the mole is large number, about 6 times 10 to the power of 23rd. So that's a lot of photons, but we can use the unit, SI unit mole, and then so that amount of photons received per square meter per second. So that's the photon flux, okay? And then if you specify the range of wavelengths, waveband, range of wavelengths, wavelengths A to wavelengths B, as the photosynthetic, wavelengths, we call photosynthetic reactive radiation, So that is 400 to 700, that range. Then you go, this is a photon flux, usable for photosynthesis. And then here is new terminology for you, photosynthetic photon flux, abbreviated as PPF. Okay, photosynthetic photon flux, abbreviated as PPF, and that is 400 to 700. But photon flux is just number of photons or amount of photons per square meter per second. So it doesn't have to be always 400 to 700. In that case, you have to call different way. Instead of PPF or photosynthetic photon flux, you might call red photon flux, or green photon flux, or blue photon flux, and then specifying this range. For example, 400 to 500. It's blue photon flux. Okay. Again, each photon has different wavelengths if you look at the natural light. And usually it's coming from 300 nanometers. I'll show you the figure in the next, maybe I should show in here. That doesn't show again. Let me, because it's not really nice. Let me quit the yeah. Let's use the PDF version to see if it works better. Lecture. Sorry about that. I didn't really realize it didn't work in this computer. There you go. Ta-da! Okay, and then I need to increase the size. So the next time I will prepare with PDF version of presentation so that you should be able to see much better way. But I need to increase a little bit. All right. Yeah, so this is a scan of sunlight, all right, solar radiation, and it shows basically the wavelengths in x-axis, and it is showing how much photon is in that wavelength. For example, so let's see, 350 or so, this sensor scanned about 0.2 micromoles. That's the amount of photons per square meter per second. And then if you look at the 500 nanometer, there are about 4 micromoles per square meter per second. So the sunlight is a collection of different photons having different wavelengths, right? And then for photosynthesis, plants use light from 400 to 700 range, okay, 400 to 700 range. So how much light, photons are available, then you need to integrate, 400 to 700 range. So you're going to find 400 about 1 point something, and 401, 1.3, 402, 1.4. So you're going to sum up in that total range, 400 to 700. And now you get the total photon flux in the photosynthetic photon flux. And then if you do that mathematically, Excel, basically summing up all the, per nanometer, value from 400 to 700, and this particular data should give you about 1,000. Oops, 1,000.
Sorry, I can't turn it off.
You might want to step out in that case from the classroom.
Sorry.
All right, thank you. So the total is about 1,000 micromoles or so in this case. Okay, 1,000 micromoles per square meter per second if you integrate 400 to 700. All right, so that's a sunlight scan. So it has a little bit of blue, a little bit of UV, and a much blue and green and red. So the three, major or primary colors. Therefore, it looks white, right? White light, sunlight. Okay. Da-da-da-da-da. So human eyes, as I said, have a different response, and then having a huge response in green, yellow-green range. Okay, so it's we can see entire range 380 to 780, but the highest response, if the same amount of photons come in, if it is green range, then we sense better. So that means we feel brighter for those colors, okay? So this is a human eye response, and then luminous intensity, lux and hood candles, based on this human response. Candela is human response. So that's why, because of this, very unique response, we are not using lux or hood candles as units to express the light intensity in plant sciences or plant horticulture, agriculture, engineering too. Okay, so that's the graph. So what is the plant response? This is chlorophyll absorption spectra. Chlorophyll A and chlorophyll B, major chlorophylls in the plant, higher plants. And you see that the peak in the red range, 600, 700 red range, and 500, 600 as a green range, and 400, 500 as a blue range. So the blue and red are the major peaks for the chlorophyll. But this is an extracted chlorophyll. Okay, this is an extracted chlorophyll. And so that means, cuvette and containing solution that is, containing chlorophyll. And then if you do the scan, which absorbance in each wavelength, then you create this kind of, response. So, but this is only the chlorophyll, right, the pigment. And then if you look at different level, for example, chloroplast or the single leaf, and express the absorption or the photosynthetic efficiency of different levels, chlorophyll, chloroplast, and single leaf, then you see that it's very different look. So this is the three pigments shown. In addition to chlorophylls, carotenoids are shown, because carotenoids, if you take the plant physiology course, you learn carotenoids helping plants to do photosynthesis, capturing light and transferring light to the action center, reaction center. So that's why carotenoids are shown in here. But carotenoids also in a little bit of green range, but not much, right? And then chlorophyll and carotenoids, it's very low, but slight absorption in the green range, 500 to 600, but mostly blue and red. And then, but if you look at the chloroplast level, then still having high peak or efficiency in the red and blue, and then start seeing green light is driving photosynthesis too. So you may have this misunderstanding of that plants don't use green light at all, therefore they look green, but it's actually not true. Extracted chlorophyll cannot use much green light, but if you look at the much higher level, chloroplast, which is basically the organelle in a cell containing lots of chlorophyll, right? And then there are so many chloroplasts in cell and in tissue and leaf, so multi-layers of light harvesting complex. Then as a whole system, if you look at single leaf response in here, you can see that almost entire 400 to 700 nanometer, which is, may not very well see, but 400 here, 700 here. So the entire range of photosynthetic active radiation is utilized. That's why we call photosynthetic active radiation as 400 to 700. Yes, question?
Is chloroplast the closest we can get to non-extracted chlorophyll?
No. Actually, we are getting chlorophyll from the chloroplasts.
Okay.
Yeah. So chloroplast contains chlorophyll, but you can actually take the chlorophyll only.
This was extracted chlorophyll. I thought you meant like you had extracted the plant, but I misunderstood you.
Yeah, actually the chlorophyll not chloroplast in the solution. Okay, so yeah, so it's very different, all right? But you can still see the little dip in the green range, and that's why it looks green, because it reflects a little bit more. So that not used meaning reflection, so that reflected, therefore it's not used. So plants look green because it's reflecting a little bit more, and then because our eyes can sense green better than other colors, so that's why relatively the reflected light contains more green, therefore your plants look green. Okay? So leaf is using a lot of green light. Interesting? Yeah, okay. All right. So this is again the same one, the leaf level photosynthetic response. Highest in the red range, and then blue is the second, and a little bit dip in the green. But 400 to 700, it's being used generally photosynthetically active radiation. Different species have different, slightly different shape, maybe a little bit lower in the blue range, maybe more flat, so different species have different. But generally speaking, this is actually many species average. This McLeay quantified and reported back in 1972, and this is called McLeay curve, sort of representing many plant species photosynthetic response. All right, red is high, blue is second, little bit less in green range. But generally speaking, entire 400, 700 is used. So that's why we call photosynthetically active radiation. But if you learn next week's lecture, then you know not just photosynthetically active radiation, but plants respond to UV light, plants respond to far red, which is 700 to 800. So outside of photosynthetically active radiation. So including that, 300 to all the way to 800 is the plant-related, radiation. Okay? So I have a name in here, plant biologically active radiation. Okay? Biologically active. So 300 to 800. Okay, so if you are quantifying, if you are engineer and designing the light environment, and you want to quantify what kind of light, environment in terms of spectrum, then you want to scan at least from 300 to 800, what's there, not just 400 to 700. All right, okay. All right, so PPF, or photosynthetically active radiation, PAR, is measured using photosynthetic photon flux, micromoles per square meter per second. And there is a sensor built for quantifying the photons, how much photons received per square meter base per second. So this is a sensor. And then, so this one, basically what it is, this is a sensor response. So sensor has filter and correction factors so that it would measure entire range 400 to 700 equally. Okay, so that's the sensor, PPF sensor, photosynthetic photon flux sensor. And then the one I have in here is the sensor, handheld. It's very, simple. So the sensor, actually sensing part is only, small. The white part you can see is sensor. Maybe I can show to the online student. So this is a sensor, right? So it's not 1 square meter, right? But it is basically adjusting the value to 1 square meter, so assuming 1 square meter range, the same uniform light intensity. But because of we are working on SI unit, we can express, micromoles per square meter per, I don't know, 0.95 square centimeter or something. So to standardize the value, we use micromole per square meter per second. Okay, but the sensor is very small. All right, so let's see. And any questions so far? Yes, Caitlin.
So when you're using a quantum sensor in like a greenhouse environment, sometimes you also use a pyranometer, which measures in 1 per meter squared. What's the benefit to measuring light quality other than one's by wavelength and the other one's by energy? Like where would the benefit be to using a quantum versus a pyranometer?
Benefit, okay, it's a good question. So actually I'm talking about pyranometer measurement a little bit later. But yeah, so what Caitlin is asking is, what is the case of using pyranometer, which is generating watt per square meter value? The other one is micromoles per square meter per second. And then beyond that, you express the spectrum using 2 units. One is micromoles per square meter per second. The other one is watt per square meter. And so it's depending on the purpose. If you want to see photosynthetic response, you've got to express micromoles per square meter per second. If you are talking about energy ratio, then simple unit watt per square meter works good. But I get that point sometime later. Okay, Caitlin. You have a question?
My question is more, can we convert from the lux or the candela measurements to a more useful watt per meter squared or micromoles?
Good question. The question was, can we convert from lux to micromoles, if there is a reporting value in lux? It's not easy to do that. Watt per square meter to micromoles is relatively easy, but lux to, I think you need to have a response curve of the luminous intensity. But anyway, the quickest way usually is if you know the conversion, lux under the white fluorescent lamp, 1 lux is such and such micromoles under this white fluorescent lamp, or 1 lux in red LEDs is such and such micromoles. So if you know the conversion, you can use that. But the conversion factor is depending on the light source spectrum. So unless you know exactly the ratio, you never be able to get the correct value.
So if we're using sunlight, then we have to use the correct conversion ratio.
Well, sunlight is not always the same, but relatively the same. So for example, early in the morning, more blue component, late in the day, more red and long wave. So it's not exactly the same, but roughly. So there is a recommended conversion. I think Dr. Giacomelli teaches that conversion. I can include that in the website, what's the conversion you want to use for different light. But again, if you want to go very accurate, then you have to have a side-by-side lux meter and PPF sensor in your hand. And then finally, you get conversion. All right, so back to PC. All right. So let's go in here. So this one is showing example diurnal change of PPF, photosynthetic photon flux, micromoles per square meter per second. It was in November, okay, sunny day. Green one is the outside greenhouse, so the natural light, sunlight. And so the peak intensity was 1400, right? And next day also 1400. So it's relatively constant as long as, the sky, the cloudiness, the same level, relatively constant. And this is a clear sky, clear sunny day, perfect Arizona sunny day. So that's why you don't see any up and down in the outside greenhouse solar radiation expressed in the ppf. The blue line in the greenhouse, the same thing for the red one. And two greenhouses have different structure, so the different structural shade, different transmission. One is heavily shaded, the other one is not. So that's why you see the big difference in the blue and red. But outside, so the incident light, original light intensity is always peak in this season, in this location, 1400, all right? But a cloudy day, much lower, right? All right, so handheld sensor like this is quite useful. And I'm gonna switch on. Yeah, so right now it's reading very low value. And knowing that, maybe right now it's a January, so the outside level is probably 1,000, in midday, 1,200, 1,000 to 1,200 in midday. And in today's outside, it's cloudy. So we're going to actually go out and see, what is the day, toward the end of the day in January, overcasting day. So we see that value. But can you guess the light intensity in this room? Micromoles per square meter per second? Midday, this season's 1,000, let's say 1,000. Sunny, outside 1,000. 350, all right? Anyone else? 200 micromoles. Anyone else? You are smiling. What do you think? Close or very different? 1,100. So it's about the same outside. All right, so here you go. This is under fluorescent light, all right? 16.6 micromoles under this fluorescent lights. It's very low, right? And when I was holding this, when I was holding this, right here, 2 micromoles. All right, human eye is amazing. Eyes can adjust to the environment, light environment. So that's why you really can't tell how much light now and outside, okay? All right, so little bit of exercise today. We're gonna just go out and then see the light level today, 5 p.m., very dark. But how dark it is compared to this room is the big question, right? Darker outside or brighter outside? All right, so let's go to the outside little bit. You can leave your stuff in here. All right, so we are outside, cloudy day.
You never know what to take pictures of.
So exciting. Right. So right now I have 40 micromoles, 10 micromoles, 28 or 80. It's changing a lot because of the clouds, I would assume. 71, 40, 32, 65. Okay, very low, right? And I don't even want it. So probably Wednesday we can, if the sun is out, we are going to measure the greenhouse light intensity, different locations under the canopy, up, above the canopy, but it's very low light intensity, cloudy day, okay? All right, so we're going to go back to the classroom and look at the photosynthetic response in terms of light intensity, so you get a little bit better sense, what means by 70 micromoles relative to 1,000 micromoles. All right? Good. Okay. Okay. Microphone still working, which is good. All right. So that quite low light level, huh? Cloudy day. And then here is the photosynthetic response curve. You have probably seen this leaf photosynthetic response curve in one of the plant science lecture or biology lecture, intro to plant and photosynthesis. It's a saturation curve. So the x-axis in here is the ppf, micromoles per square meter per second. You know, when you first saw this kind of graph earlier, you know, before you took this lecture, you probably didn't pay attention to the unit, right? Now, look at the unit. It's shown up to 2,000 micromoles, okay? So the summertime midday, we get this light level, 2,000, a little bit beyond 2,000 micromoles per square meter per second, ppf, midsummer day, okay? And then November, what was the level? Like a 1,400, right? And then now it's probably 1,000 to 1,200-ish, the light intensity. So, so the photosynthesis, this is 3 different conditions. Don't worry about the conditions. I'm showing just the real data obtained for tomato plants. And then it's saturating, right? So it's increasing by increasing photosynthetic photon flux, light intensity initially, and then start slowing down, and then saturate, right? So any more increase wouldn't contribute, you know. So that's why we say saturation curve. Look at the light intensity close to 100 micromoles or so in this one. It's almost 0 photosynthetic rate. Photosynthetic rate is expressed as CO2 absorption, and then again used mole. So the micromoles of CO2 absorbed per leaf of the plant per second. So it looks like the same unit is used, right? Micromoles per square meter per second, but the meaning is different. So the y-axis is the CO2 absorbed or fixed per square meter of leaf per second, right? And then the x-axis is a ppf, so micromoles per square meter, you know, of the surface, photons received per that surface, in this case leaf per second. But it's a per square meter of surface, you know, the light intensity. So about 100 micromoles or so, net photosynthetic rate becomes 0, right? And below that, usually you have more respiration rate. Remember, plants do photosynthesis and respiration at the same time? And then depending on the light intensity, photosynthesis is much higher, or respiration becomes relatively higher than photosynthesis when very low light intensity is available. So that's here. Okay? So what we have seen outside was what? It's ranging very quickly, but highest light intensity was 70 micromoles we recorded outside, right, today. So it's very, very low, right? If you have plants in that cloudy 5 p.m. situation, they're not doing much of anything. Photosynthesis, no, it's not much. Here, inside the classroom, 2 micromoles, no way, right? So nobody is even trying to grow plants in this classroom because you don't get that high light intensity, right? So that's why you are suggested to put your plants close to the window if you need to keep the plants inside, right? Rather than, somewhere away from the window. Even, window side, it's very low. And then maybe we can demonstrate that Wednesday, how close to the outside the window side is in terms of light intensity. And it's usually very, very low compared to the outside. Oh, yes?
So when you're growing anything under artificial
Yes. So the question is, if you are growing plants under artificial light, then are we able to adjust the light intensity? The answer is yes. You can do two ways: increasing number of light fixtures, so more lights, or use something dimmable. Then you can dim or increase the light intensity. Usually changing either voltage or what we call duty ratio to change the output of the light fixture.
Would you want to mimic the natural?
Oh, do we want to mimic, or can we mimic the?
Is that preferential to mimic?
So for example, if you are applying very high light intensity in artificial lighting, it's a very good question. So starting from 0, jump to 1,000, 2,000 if ever possible. It may not be a good idea because it's a huge stress to the plants from 0 to that high level. So you might want to ramp up the light intensity if possible. But most of the indoor growing, the light intensity is about here, 500 or even less. So somewhere between 200 to 500 micromoles is a typical light intensity in artificial or electric lighting, indoor vertical farming situation. And in that case, it's not a big jump. So it's not so much a problem to jump the light intensity from 0 to 200 or 0 to 500. So plants can take that. The reason they use relatively low light is because it's very expensive to generate that high light intensity. So that's practical reason. But instead of having big peak in the middle and then sort of, the sine curve type of situation, in artificial lighting conditions, we set constant light but long photo period. So that as a total energy, it's maybe equivalent or close to the natural light environment. So that we talk about that more later, okay? Yes?
If you were adding more lights to increase the intensity of the light, would there be a point where adding additional lights wouldn't increase the intensity, or could you basically add an infinite amount of lights, depending on the size?
Good question. I'm gonna hold because I'm answering that question. The question is, it's beneficial to increase the intensity forever, or there's a point, So if you look at this curve, there's a saturation, right? So that means if you increase beyond, in this case, 1,500, there's no use to increase to 2,000. It's the same photosynthesis. But this is a leaf response, single leaf, okay? And then I'm going to talk about more complex canopy, whole plant canopy response. Okay, it's very different. Okay, so that's super important question you asked. Yep. All right. Okay, so this is the definition of net and gross photosynthesis. If you're not familiar with net photosynthesis versus gross photosynthesis, this is, you want to read what is net photosynthetic rate, how different from gross photosynthesis rate. The gross is the true rate of photosynthesis. And if you consider gross minus respiration, that's net photosynthetic rate. Because again, photosynthesis always, happens when respiration is happening. And respiration is basically opposite, reaction, releasing CO2, and photosynthesis is, absorbing CO2. So when you measure, what you measure is a net photosynthesis. Net photosynthetic rate is actually assimilation. So that's the gain, right? Gain of carbon absorption. All right, so you read this, okay? All right, and then the net photosynthetic response of the leaf is characterized using this simple chart I created, and then having 3 different characteristic values shown in here. One is the maximum net photosynthetic rate. Leaf photosynthesis, particularly what we call C3 photosynthesis. There is C4 and CAM, but C3 is the kind of photosynthesis for most of the greenhouse crops. So usually don't talk about C4 photosynthesis in my lecture, but C3, so this is a major type of photosynthesis for horticultural crops. It's saturating, just like you saw. And then, so that saturating meaning there's a maximum level, right? It sort of plateau, right? So that's a maximum net photosynthetic rate. And then another parameter you can use to express this, saturation curve of photosynthesis, the light saturation point. That's not the photosynthetic rate, but the light intensity. So the light intensity that is causing near saturation or saturation. All right, so that's the saturation point. Light intensity that saturates net photosynthetic rate. Light saturation point. And then a light compensation point, and that is the intensity where photosynthesis, gross photosynthetic rate is balanced with respiration. There are two kinds of respirations if you know plant physiology, dark respiration and photorespiration, but generally, as a general, it's a respiration. So photosynthesis balanced with respiration, that light intensity is light compensation point. And light compensation point is about 50 to 100 micromoles, which is shown in the next slide. But so that low light intensity, almost like outside today, that is a light compensation point for most of the plant species. We work in horticulture, in greenhouse. And then this initial slope, so that is the slope or increase of photosynthesis when light is the only limiting factor. So it's almost linearly increasing, right? Linearly increasing. And that slope is called quantum efficiency. And that is the true efficiency when light is the only limiting, all right, of the photosynthesis. Okay, so there are several things happening here, okay? All right, and then, so this is a summary of the value range. So the maximum net photosynthetic rate for greenhouse grown plants, vegetables, usually somewhere from 10 to 20 micromoles. You don't need to remember this, but 10 to 20 micromoles of CO2 absorption per square meter per second. It's a leaf level, okay, leaf level. Light saturation point, 1,000 to 1,500 micromoles per square meter per second PPF, photosynthetic photon flux, which is 400 to 700 wavelength range. Compensation point is about 50 to 100 micromoles. So that's sort of, giving you the picture of what is photosynthetic response. And this is actually the reported value, tomato, cucumber, watermelon. And those values are affected by growing conditions and in species and maybe cultivars too. So, so it's not always the same, but you can see the relatively, tomato high compared to other crop species and things like that. And this is one example how growing conditions affect that, saturation, light saturation curve of photosynthesis. Highlight grown plants, usually having a much higher maximum photosynthetic rate and then also saturation point. Low light growing plants, even in the same cultivar, in the same species, when they adapted to low light, then they would reduce the maximum photosynthetic rate at saturation point, but increase the initial efficiency like that. And seeing that more or greater photosynthetic rate under low light range compared to highlight grown plants. And then next slide is showing the actual data we obtained for the same plants, top leaves, which is adapted to highlight, and lower leaves or the dark grown plants, more adapted in a low light environment. Yes? Probably not. Yeah, so I'm trying to be careful to explain this, but what you see in the tomato crop is the difference between the growing light environment, top and then below. We will hopefully quantify that Wednesday. And the plant age, leaf age. So the top leaves are relatively younger, and compared to very low, down below is a more aged senesced. Not quite, but different age. So the age, physiological age, affects the photosynthetic capacity too. But I don't think it would show this kind of high efficiency under low light. If you see that this is not the age effect, it's more like a low light adapted response. Because growing under low light, plants try to make the leaf thinner, and you actually have a different structure in the leaf and capturing more efficiently the light. So that's what the difference. And then I believe those are growing in a different greenhouse. So that's why I included in here. So they're not including age effect. So so this is the actual data of tomato plants grown under low light and grown under very high light. The leaf response, the difference. All right, how am I doing? Okay, so I have several other examples here, using system like this. This is a portable leaf chamber measurement system. So we can sandwich the leaf and measure the photosynthetic rate. And then it has a lighting system, and then I can punch in the number I want, like a ppf, a photosynthetic photon flux, and then measure the photosynthetic rate. Very handy, just showing. So we will be talking about plant canopy situation, and then I'm going to show you, I'm going to review this next week, but here is the most important graph of those two lectures focusing on the light intensity. The difference between leaf photosynthetic rate, so the light response, again saturation, and canopy photosynthesis, which is more like a linear, in the same range of light. The unit, so maybe this is a sort of unofficial homework, but think about why and what is causing this difference. And then we talk about Wednesday. All right. So one is saturation. It's a leaf, single leaf of the same crop. This is, I think it's tomato. And then the unit, for whatever reason, they use watt per square meter instead of micromoles. But here's a conversion of this light source. I know 1,000 micromoles ppf is about 200 watt. So they got good, like a winter midday light intensity. And then leaf is saturating, but the canopy, which is basically the number of plants growing, like in this situation, this is not the tomato, but in that case, if you measure the whole canopy photosynthesis, it doesn't saturate. So the big question I want to ask you to think about why this is happening like this. All right, so again,